So it's really a pleasure to be here today talking about our latest work that was published earlier this year in Science Advances. The paper was spearheaded by a collaboration between my group at Rockefeller University and a fantastic proteomics group at SciLifeLab here just across the street from where we're talking right now. This paper, the first author is Ilana Kotliar, a graduate student in my lab. And she was asking the question, what interactions are important to regulate the function of a class of receptors called G-protein coupled receptors? These are found in all cells in the human body, but it turns out that their surface expression and maybe their pharmacology is regulated by another ubiquitous group of proteins called Receptor Activity-Modifying Proteins, or RAMPs. Now, the existence of these two proteins has been known for decades, but just recently there was a hypothesis that their interaction was much more widespread than had been previously noted. So Ilana had the idea that we could develop a multiplexed method to look at all possible interactions between about 250 GPCRs and the 3 RAMPs. There are only 3 RAMPs. There are 250 GPCRs. What are the pairwise possibilities for their interaction in cell membranes? So in collaboration with SciLifeLab, we developed a strategy to look in cell membranes at those interactions. And that technology is a multiplex technology based on immune capture with specific antibodies against the GPCRs, the RAMPs, and against engineered epitope tags that we put onto the GPCRs and onto the RAMPs. And this is where the collaboration with Atlas Antibodies was very important, because we used antibodies from Atlas that were GPCR specific that we had studied earlier in previous work. So that is basically the essence of the question. Now, to answer that question, to address the question, Ilana expressed all possible combinations of 220 GPCRs and the 3 RAMPs. And then we did this in New York. Then she brought these samples to Sweden to carry out studies at SciLifeLab using their Luminex technology, which is a suspension bead array assay, which is a very cool technology that involves the ability to couple an antibody to a bead that is IR coded, IR dye coded. There are about 600 possible beads, each with a different color. So you can put up to 600 different antibodies, each antibody on a different colored bead. And then when you combine all of these samples and mix them with the beads, you can pull out receptors or RAMPs or receptor RAMP complexes and interrogate that mix in a flow cytometry kind of a setup. And that's where the group at SciLifeLab really helped. The amazing result of this work was that we found that about 120 of the 225 GPCRs actually interacted with at least one of the 3 RAMPs, and some with more than one of the 3 RAMPs. So this was, I think, very important work, because in the past, the field advanced just iteratively, step by step. If you had a receptor that you were interested in and you wanted to see if it interacted with a RAMP, you could study that one by one. But now we've studied 200 plus receptors with all 3 RAMPs in one experiment. And that result of that is that we have a complete understanding of the interactome between GPCRs and RAMPs. That's basically it. Well, some of the innovations and technologies that we use for this project I think are very worth mentioning. One is the ability to engineer receptors that have antibody epitope tags on the N-terminal tail and C-terminal tail. That's very important. That's pretty standard work nowadays, but what's not standard is selecting the correct monoclonal antibodies and knowing the epitope and the location of the epitope to optimize being able to detect them later in the suspension bead array in a flow system. Secondly, we developed a technology to validate the specificity of antibodies raised against the receptors themselves. These were the Atlas antibodies from the Human Protein Atlas that we mainly analyzed. So the ability to validate whether or not a specific antibody binds to its target receptor, but not to all of the other 200 receptors, which are very similar in sequence, was important. Finally, the suspension bead array approach is very important. This is the so-called Luminex technology that depends on these magnetic dye encapsulated micro beads. So each of the beads is embedded with an IR dye, which can then be decoded after you have a binding event in a flow cell. This is an important technology. This was pre-existing. But then, once you do your suspension bead array and get the multiplexed output of that, in order to say that you have a specific GPCR interacting with a specific ramp. For example, the GLP-1 receptor interacting with RAMP1. You have that in a flow system where you overexpress those two receptor ramps in a cell and then pull it out of the cell for the analysis. But what about the reaction, the interaction in the cell itself, in the membrane, unperturbed by detergents or extraction procedures? And could be in a cell line or it could be in a primary cell. How do you determine that? And I think that's another important technology that we used. And that technology also from Atlas was this MolBoolean proximity assay, which for us was very important because we had to validate that the suspension bead array results actually were recapitulated in the cell membrane. And this assay takes advantage of antibody binding in cells, and if the two targets of the antibodies are in close proximity, then you get a fluorescent readout. The advantage of the MolBoolean method is that you can actually quantitate not only the complex formation, but the individual components independently of the complex formation. I think this is really the advantage of the MolBoolean. It is a type of proximity assay, but it goes way beyond these just colocalization type of confocal colocalization experiments that have been done for decades. I think it's frankly revolutionary because you can do it in cell lines. You can do it, of course, in cell lines when you overexpress the components and create controls, but it's also compatible with primary lines, which we've done. So our goal now is to validate, using the MolBoolean strategy, all of the screen hits that we got from the suspension bead array assay. Well, so you might be asking, how does this work have some potential impact on medicine? How can it help people who have diseases, maybe for therapeutics or even diagnostics? And as a physician, by training, I want all of our work to have some disease context. And I think this work is a good example of how, at a very early stage, we're developing technologies that could be used later in medicine or precision medicine. The key here that we're talking about is this concept of protein-protein interactions. There are thousands, literally thousands of different kinds of protein in a typical cell, not only inside the cell, but on the cell surface. And proteins interact with each other. They bind to each other. They interact transiently. They may form tight complexes. The receptors on one cell surface may bind to proteins that are secreted from another cell. And all of that is important for the physiology of the organism. And when these protein-protein interactions get dysfunctional, that could cause disease. I think that one of the important things in what we're talking about, these interactions between RAMP proteins and GPCRs, is that it's very important that the GPCR in most cases is at the cell surface. Without the RAMPs, some of those proteins don't get to the cell surface, and then one cell can't signal another cell. And when cell signaling is disrupted, you could have effects on metabolism, or you could even have diseases like cancer, which are caused by disruptions of cell-cell interactions and cell signaling pathways. So I think being able to measure the interactions between proteins in cells in a quantitative way is very important for the future of drug discovery.